and if I want to do a quality control more specifically, I have to choose if the monomorphic markers in one heterotic group will still, or I will remove, or I will keep in both heterotic groups. So many questions about this quality control, so I prefer to use directly all the markers that he presented or published for do this analysis. But of course, it is possible to do different quality controls to check what is going on with the results.
Yeah, I wasn't really hinting on that. Maybe the results would be different, but as far as I understand it, a minor allele frequency threshold is done to, you want to avoid that you keep SNPs that seem to be segregating, but they're not just the genotyping errors. And if that's the motivation, then 5% is pretty high. I mean, if you have a few thousand animals, then 5% means probably that you see hundreds of the copy of the minor allele, right?
Yes.
So then I would rather prefer to say, okay, I want to see at least 50 copies, and then I determine my minor allele frequency based on that, considering the size of the population.
Yes.
Okay. You also did some edits based on Heidi Weinberg equilibrium, and this is on, I guess, it's in your chapter 2 on page 38 of your thesis. But so I wasn't sure how severe this edit was, because you do not describe how many SNPs that do you drop in each of those steps. So I wonder, do you have a feel if you use this p-value cutoff of 0.05 or?
I didn't present, but I know that, for example, for the math to this threshold of 5%, it was around 2,500 SNPs that were discarded. And for the Hardy-Wilmer equilibrium, it was like 500 SNPs.
500. Okay, so that's not so many. Do you have any feel for what kind of SNPs that? Well, they are not in Hardy-Weinberg equilibrium, but how severe is that deviation? Is it? Do you have a feel what are the properties really of those 500 SNPs that you removed there?
This is because, of course, because they are deviated from the threshold of the Hardy-Weinberg equilibrium. Because maybe most of the markers, or some markers, of course, are not following the Hardy-Weinberg equilibrium. But I couldn't get exactly your point of your question.
Yeah. So is it really SNPs where you, for instance, you see only 2 of the 3 genotypes? Is that how extreme this cutoff is? Or is it also those that, you see all the 3 genotypes, but the frequencies, they just deviate from Hardy-Weinberg?
Yes, I think if they are deviating, so it will be, we don't obtain the expected proportion of the genotypes in the population.
Okay.
Yeah.
So then maybe let's move on to some of these results in this chapter. So looking at table 2, page, maybe one other question first on your models. On page 39, you say that for model A you did not include the inbreeding depression parameter. And I was wondering, you just make this statement, but you don't explain why you did not do that.
So you mean in the chapter 2?
Chapter 2, page 39, the left column towards the bottom, the third, fourth sentence from the bottom, it says model A did not include the inbreeding depression parameter.
Page 39. It's for estimation of the marker effects?
No, it's model A. The model where you use pedigree only, if I understand correctly.
The other copy, I think.
No, but here.
Yeah, sorry, I'm working on the copy that was sent to me some time ago, so I'm not sure if there's a newer one, but.
But sorry, I couldn't get the line that you say.
Well, the statement is that model A, so the model where you fit pedigree, you know, where you fit the pedigree, not the snap effects.
Ah, sorry. Okay, the model A for the pedigrees. See, I didn't include because then I had to calculate the breeding coefficient from this model, and normally in the models, in the crossbreeding, in the picture breeding, there I'm not using this effect of genomic inbreeding in the pedigree model.
Right, so you just didn't do it because that's what conventionally has been done, if I understand correctly. Then moving on to the results there in table 2, you have these variance components estimated. And just looking at these estimates, only focusing on the variance components in this table 2, I would advocate we should go for model A, because it has higher genetic variance, it has a higher heritability.
Yes.
So my question is, would you agree with my statement?
If I will be agreed with your, sorry?
With my statement that based on this results in this table that model A is preferred.
I would prefer the genomic model because it's more accurately for the estimation of the variance component. But also here I use it, I trace back the pedigree only 3 generations.
Okay, so then maybe can you try and explain to me why are the additive genetic variances with the pedigree model higher than with your genomic models?
Because here we were using just a sample of the purebred individuals, so maybe not the genetic variance will be reduced because it's a sample. In the case of pedigree, it would include more number of
Mario, you're fine.
I'm sorry, I don't know what's going on, but
I don't know. David, do you hear us?
I cannot hear you.
David, Andres, do you hear us? Apparently we were disconnected. David, are you here? Can you hear us, David?
No, está bien.
We can see you, but we don't hear anything from you.
You can hear me?
Yes, that's okay now.
It's okay now? Okay.
Mario, is it okay for you?
Yeah, for me it's fine, yeah.
No,
Okay.
so now everything is back. The point is that I muted the audience, and when you mute the audience, you need to unmute the whole audience, not one person at a time. So it was my fault. Sorry about that.
Okay, so we were at that table 2, the additive genetic variances with the model A, they are higher than with the other models, right? So
Yes.
you say something about sample size, but I believe that you use the same phenotypes across all the models, right?
I did the same, but the same animals, but the pedigree was, I traced back to 3 generations to estimate the variance complete. So maybe it could be the difference. And in the case of for estimated from the genomic models, I use only the animals that were genotyped.
Okay, so the additive genetic variants we're looking at between the pedigree and the other models, they pertain to a different base. Is that it?
Sorry, I couldn't hear you well.
So if I understand correctly, the additive genetic variants between the pedigree-based and the genomic models, they refer to a different base. Is that what you're saying?
I mean, I use the same animals with records, but for the pedigree, to construct the pedigree, I trace back to 3 generations. So
Yes.
probably this could be the reason why the additive variance is different.
So but let me try and rephrase the question. So what is the base to which, or to which generation do these estimates apply for the additive genetic variants based on the pedigree?
Sorry, I couldn't get your point of your question.
Can you not hear me, or is the question not clear?
The question was not clear.
So if you use pedigree, you have a relationship matrix which has a certain base, right?
Yes.
And the variance that you estimate, that applies to that base of your relationship matrix. And the same applies to your genomic models. So I was wondering, is there a difference in base between those different relationship matrices which can explain the difference in estimated variances?
Yes. Yeah, I cannot. I don't have the answer now.
Okay. Because if that would be the sole explanation, then I would still expect the residual variances to be the same across the models, but that's also not the case. So for instance, in the first line, it seems that with the pedigree, you move away variance from the residual to the additive genetic variance relative to the genomic models, right?
Yes.
And so then the question is, of course, is the pedigree model overestimating the genetic variance, or is the other one underestimating it?
Could be, yes, could be, but it will be difficult to know if it is due to this reason, I think. Because, for example, in the case of the average piglet weight, this trait that include the permanent effect estimated of the additive variance between the pedigree and the genomic models were the same. So it could be, yes, that pedigree could be overestimating the additive genetic variance.
Okay, yep. So maybe let's move on to chapter 3. There you have simulated different scenarios with different variance components for the additive and dominance variances. I was just wondering, how realistic are the different values in terms of the amount of dominance that you have simulated?
Yes, I tried different level of ratio of dominance to additive variance. For example, I tried extremes to see how it is working, the mate allocation and the breeding scheme in general. But yes, like we said, in real, this proportion will be low than the one I tried. So in my case, it was with the objective to try a string to see how mate allocation is working.
Right, so would you say that the first case that you consider, so when your heritability is 0.1 and the dominance heritability, as you call it, is, or the HD squared is 0.01, is that closest to what we would observe in reality?
I think the most close is the case 1, the first one, with heritability equal to 0.1 and 0.01, because this is the one that I use from, I took from the paper of Shang et al. 2016, real Landras and Yorkshire population.
Okay, yeah. Then you simulate heterosis, or heterosis sort of arises as a result from simulating dominance, and you also you estimate heterosis, if I understand correctly, as a function of of dominance, or at least of heterozygosity or homozygosity, right? In your models, yeah.
I simulate the heterosis as directional dominance.
Right.
Also, yes, following the dominance, directional dominance.
Yeah, and then in the models here, you estimate it by doing a regression on heterozygosity or homozygosity, right?
Yes.
Can you tell me what is the classical definition of heterosis?
The classical definition is the difference between the average performance of the parents and the performance of the crossbred individual. This difference is the classical definition.
Right. Yeah, I agree. Very good. So what if you had used that definition to estimate heterosis in your models? Would you expect that you had gotten the same results?
It will be, yes, I will say I did, but we use this way that is, I will say this is suspected heterosis based on directional dominance or model like Falconer, that is difference between allele frequencies by the product of the dominance effects. This is like the theoretical or expected heterosis. I use this way because I modeled the heterosis like dominance. So I prefer this, but yes, it can be possible, of course, to use the true heterosis or the real heterosis. And you can hear me? Yes?
Yes, yeah.
And yes, I did, for example, and yes, there is heterosis, the true heterosis that you asked me, but in one of the scenarios, for example, for scenario 1, I obtained a negative heterosis that was a bit surprising in this case, yes. But yes, of course, it is possible to obtain this true heterosis.
So if heterosis is proportional to heterozygosity, because it's, let's say, due to directional dominance, then my expectation would be that across generation, in absolute terms, heterosis does not change. So would you agree with that, or?
I think heterosis will be changed because it will depend in the difference in allele frequencies in both populations. So if I do selection across generation, the allele frequency will change, so heterosis will change.
But is that change, is that directed, or is it just sort of random that it can fluctuate, or will it go up or down? Or?
No, I think it is directed by selection. One of the slides I showed that selection for crossbred performance resulted in higher heterosis, expected heterosis, than selection for period performance. So I think it's not something that is at random.
Okay, but that was in one of the slides that you showed, right? I did not recall that I had seen that figure in your thesis.
Yes, I didn't present in my thesis, but I construct this graph with the numbers that I present in my thesis.
Right. Okay.
It's the same number, just in a graph.
Yeah.
Okay, Mario, one last question, maybe.
Okay. Yes. So let's assume that. Are you still there? Yeah.
Yes.
Let's assume that, I don't know what you're going to do after your PhD, but as of tomorrow you are the manager of a breeding program of pigs.
Okay.
And then, which means that every euro that you spend in the breeding program, you are responsible to make some returns out of it, right? So based on what you've learned from your thesis, what is the first thing that you're going to change or implement?
I would like to say to use genomic selection for crossbred performance, but the thing is that we will obtain, for example, a reduction in the genetic gate in the period performance, and in real pig breeding it is important. So first I will do extra analysis, for example, to trade a combined index to balance the genetic gain across purebreds and crossbred individuals before to implement this technique, for example.
Okay, yep, thank you.
Okay, thank you, Mario. I will now give the floor to Aaron for questions.
Okay, can you guys hear me?
Yes, I can hear you.
Okay. All right, thank you very much. And nice presentation, David. Nice dissertation. I enjoyed reading it very much. Thought it was well conducted, well researched, well referenced. So I congratulate you on that.
Thank you.
So I just have a few questions, mostly focused. I mean, since Mario covered pretty much everything else, I can cover, I think it's chapter 4, the one with the maze single crosses, which I'm best prepared to ask questions on. So, I'm guessing, one main objective of this chapter is to define models that better separate out the genetic components in a population of single crosses, right? So I just had some basic questions about the correlation between these different matrices. So I think you said you correlated the matrix between the additive genetic relationship matrix with the epistatic relationship matrix. Did you look at any relationships between the dominance, how you derive the dominance relationship matrix with the additive genetic relationship matrix? Were those also correlated with each other?
Yes. It's true, yes.
They were, okay. So I guess, so then, I mean, the limitation, so there's still some linear dependencies there between these different matrices. So you think this is something that is possible to get around statistically, or do you think that is possible to separate out these components. To better separate out these components, you need to derive different populations.
Sorry, you mean I didn't get the last point of the question.
Well, so I think one of your objectives was to better separate these components of variance, right? By better formulating different matrices. But I mean, would you say that you were completely successful in doing that, or unsuccessful, or partially successful?
Yes, I would say that this is it's bringing new light to how to model the hybrid genotypic values, and we split the components. For example, in the GCA we split it in additive and within line epistasis and also dominance. But for example, you saw in the results that I presented that there is still some confusion with I estimate the dominance and across population epistasis. But this is something that for the moment I think we have to do this, because non-orthogonal models, for example, are good, but they are full of some assumption. For example, it is Hardy-Wimber equilibrium, and the hybrids, of course, there is not a hybrid Hardy-Wimber equilibrium, and it can lead to this confusion. And also because the linkage disequilibrium, the model assumes that there is linkage equilibrium, but in reality, and especially in these populations, we know that there is a strong linkage disequilibrium. So this can also to be difficult to estimate and separate completely both dominance and epistasis across population components. But I think it's better that using a model that is non-orthogonal.
Mm-hmm. Okay. So what, I mean, if you really wanted to get to the bottom of these components and estimate them perfectly, what kind of population would you develop?
So it could be F2 population, that this is expected to be in Hardy-Wimber, because the ratio of the different genotypes is according to the Hardy-Wimber. So it could be a population that we can better partition between the components. Yes, I think our mapping population, it's called sometimes in the literature.
Okay. I mean, there's still LD in an F2 population, though, correct? It is expected at least to be less than in hybrid populations. So we need to do many generations of random mating to break down all the linkage disequilibrium. Mm-hmm. Okay. And so that then along the same lines, so did you look at the correlation between your additive relationship matrix and your formulated, the way you formulated your dominance relationship matrix? What were was the correlation between those two matrices?
You mean between the additive and dominant?
Yeah.
I think I didn't include in the paper, but it was quite low, the correlation between the additive and dominance deviation.
It was, okay.
Yes, it was quite low.
Okay. One thing I noticed in your results is that using your matrices, you found that the within group additive genetic variance was higher than the between group. The within group additive epistatic variance was higher than the between group additive by additive epistatic variance. So that, do you think that those differences, I guess, are real? And does that surprise you? Which would you expect to be higher?
One would be expect that the across population, that is more related to specific commanding ability because heterosis, but in general, in this, for example, in this kind of population that are divergent population, the specific commanding ability tends to be lower than the general commanding ability. So I think if the general commanding ability tends to be higher, then the within line epistasis, it will be even a bit higher than the across population epistasis, yes, it could be possible.
Okay. So you're, I mean, like a lot of other studies, you found that the total proportion of specific combinability variance was fairly low relative to the general combinability variance. Why is that?
Because when we are selecting by reciprocal recurrent selection, I think most of the genotypes that produce most of the loci with dominance or over-dominance, for example, will be fixed in both populations, and it will reduce the dominance variance or the specific combining ability will be reduced in this kind of population, and most of the variance is explained for the general combining ability.
Okay.
This is in one paper from Rafe 2007, I think.
Okay, good. Just thinking about my train of thought here. Just the accuracy of estimating these variance components. And so you use a real data set to estimate these variance components, and you claim that one model is better than the other model for estimating these variance components. You claim that the model that includes basically the residual unrelated effects, kind of that catch-all, that will then bring down the bias of the overall additogenic variance as a better model than just the more traditionally derived one based on pedigrees, not including that effect of the model. So I mean, but I guess I'm these are real data, and so I guess how do you know that's true? Because your model fit criterion are all pretty similar between all these different models. There's really no big difference. So I mean, would it be possible to come up with a way to really determine whether or not your model more accurately estimates these variance components?
Yes, it will be, well, I would say possible doing a simulation. At the moment, we don't know how to simulate epistasis within population and epistasis across population. So it's something that is still not clear to simulate and see if the model is more accurately. Simulation is one way, but we have to think first how to simulate epistasis intra and across populations.
Okay, so you're working on that. So okay, well I think that'd be something interesting to look into to see if you can accurately simulate those effects to see whether or not your models better capture those. If you have a simulation to tell you what the real answer is. So then going to the genomic selection application of all of this. Your prediction accuracies at the end of the day were pretty much the same, right, between all these different models.
Yes.
So I guess I'll just ask you again, why is that? Did you expect that, or was that unexpected?
Yeah, it was the accuracy was almost the same in the different models, and it was quite expected because, as we discussed before, most of the variance is due to the general commanding ability. If the specific commanding ability is quite low, then it don't contribute too much to the prediction accuracy. So it was quite expected in this population.
So what about different scenarios of hybrid single cross prediction? So you just took the whole data set and divided it up and did a pretty simple 5-fold cross validation. What if you looked at different scenarios of predicting the single crosses? Would you expect a different result or the same result? I mean, you should. I mean, in theory, you're getting a better estimate of the true underlying additive value, and so I think that has some sort of application in the context of genomic prediction.
Yes. In this case, I did 5-cross validation. That is like a mix of for example, the cross validation that Techno used that include T1, T2, and T0. And we did use 5 cross validation in order to compare the models under similar conditions. But yes, it can be done in another cross validation schemes. In fact, I would say that I did following this this cross-validation, and the results were the same. So within each validation sets, for example, T2 or T1 and T0 was the models performance equal. I don't observe any improvement by using a full model, for example, including all the non-additive genetic effects. But it will depend also on the population that is under evaluation. Maybe in another kind of population, we will see some difference in between models.
Okay. Okay. I'm going to look at my notes here. So you didn't, so correct me if I'm wrong or not, but I mean, you didn't really see much of a difference when you treated the allele effects differently, right? Like you assume the allele effects, if you derived a model that assumed the alleles came from different populations and had effects that were different between those different populations, there was no difference there?
In terms of goodness of fit of the model, and in terms of predictionability was the same. So I think the models are equivalent. But in terms of partition of the genetic variants, so it's completely different.
Yeah. Okay. Okay. I guess I'll ask a question similar to what Mario asked, in that if you were tomorrow ended up being the lead of a corn breeding program someplace, how would you use the results from this work that you conducted on maize single crosses to motivate perhaps the redesign of breeding operations? Have you given that any thought?
Yes, I would say I would use the full model, the recommendation if I am the manager of the breeding program. I would use the full model, also including the remaining related effects. In order to not overestimate the additive genetic variant that will have consequence, for example, to overestimating the genetic progress that I will obtain at the end of the breeding program. But also, of course, more research are needed to evaluate these models in maize data sets to see how it's going with this model in other data sets. And if it works properly, then I can try to implement in my breeding program.
Yeah. Okay. Well, let me push back a little bit. So I mean, but the genomic prediction accuracies were the same between all these models, and so I guess you make the case that it'll allow you to better estimate the additive genetic variance and perhaps allow you to forecast genetic gain into the future. But I mean, do you think that's really important for a breeding program to do that, to spend, or are you better off spending your time and energy just trying to make predictions and go forward for the time being? Is it? Yeah. How important is it to really get a handle on that additive genetic variance for the purposes you described?
So I will use the more simple model with just additive effects to predict if my object is predict the hybrid value. I will use the most simple. But for selection of inbred line, I will do more research within my model to see how is change the ranking of for selection of inbred line, including using the full model or the related remaining effects. But for hybrid prediction, which is my objective, I will use the most simple one.
Yeah. So at some point in your dissertation, you mentioned something about having a better handle on these genetic components could allow for better training population optimization. If I'm correct in you mentioning that, can you elaborate on that a little bit? I mean,
yes. For example, it could be, I think, interesting. Now we have the coefficient for dominance deviation. It could be also interesting how to for optimization of the training set for calibrating the model. Also, it will be interesting include the dominance effect to construct or to optimize the calibration set. It could be interesting to research.
Yeah.
Yeah.
I mean, you haven't looked at that yet. Have you looked at that yourself? Have you looked at that yourself yet?
Sorry, I couldn't.
Have you looked at that aspect yourself yet, or not?
No, not.
Not yet. Okay. I think that'll end my questioning.
Okay. Thank you, Aaron. I will now give the floor to Leopoldo.
Yes, can you hear me well, David?
Yes, I can hear you, Leopoldo.
Okay, first of all, thank you very much for inviting me for this dissertation and to having the possibility to read your thesis. I think, being a tree breeder like me, between pig and mice breeders is an honor. So this thesis, I think, is really very well written, very didactically. I think the thesis presents advancements in such a clear, such a simple way that one wonders why this has not been done before in such a way. Anyhow, I will have a few comments on a few aspects that I think that could be somehow perfectible, and then I will go on with a few questions. So the first thing was that being meta location a kind of important concept in the thesis, it was a little bit surprising to me at the introduction level to the little place you give to it. So anyhow, I mean it's a simple concept, but I was wondering whether there were more references out there concerning this aspect. And then there was also another aspect concerning also this meta location that at the beginning was a little bit confusing for me, coming from external observer, that meta location was not clear to me, the difference between that and mate selection, for instance. And I was reading the document that I got at the end. It was something like at the page 130 something, that there was written the difference. Of course, reading the document, I got the difference, but it could have been nice to have this clearly set from the very beginning. So, this is just a general comment. Otherwise, I think it's really very, very well written and clear. So, I don't know if you want to comment on this comment before the question, David.
Yes, I get your points for your comments from my thesis. Thank you.
Okay. Yes. Then my first question concerns this pre-selection. You did before the meta location. Of course, the meta location, it consumes a lot of resources. It's an optimization, and then, of course, you need to do this pre-selection in order for it to be simplified somehow. So I didn't get really the point with the different scenarios you did in the appendix 2.2, where you try to see to what extent pre-selection was important or not. Of course, I mean, the point is that this pre-selection does not affect the final result. But could you explain very briefly the different scenarios you choose to in order for you to set that pre-selection was not affecting the result?
Yes, of course. In the case of the preselection, one of the main conclusions of the study is that the maximization on the total genetic value don't result in a reduction of the additive genetic gain. So one question was, maybe we don't observe any reduction in the additive genetic gain because we did a preselection of mates, and we are sure that we are keeping most of the additive genetic gain because, of course, the mate has the higher selection intensity. So, and it was also one of the questions of the referees. And what we did to discard the influence of the preselection is that we compare another two scenarios. For example, just selecting the 40 males, that it will be like a selection standard selection truncation, and the other extreme was select the 120 males at random to see what happens if the genetic gain is still being the same or it will change. So the results at the end show that the additive genetic gain is the same. So don't select maximization on the total genetic value doesn't have a detrimental effect on the additive genetic gain, independently of the preselection that we can do in the for mate allocation. I don't know if it is clear for you. This.
Okay. So well, ultimately, the best solution could be to do some kind of mate selection in order to check whether you really get the best mates out of that. But of course, this is unfeasible because you have so many selections and mates to do. Okay.
Yeah,
Well, okay. Another question it's concerning this inbreeding effect. So of course I think that's a nice feature that is not always done, at least on my field. This adding this covariate for inbreeding as a in the model. A question I've got when I was reading, and then at the end in your final discussion, you gave a comment to that, is that of course this relationship between let's say the breeding and the changing mean, it's usually linear, but when it comes to epistatic effects, this could be non-linear. Did you check any quadratic coefficient in the regression together with the linear regression in order to see whether this quadratic was also important?
Yeah, no, I didn't check, but that's true that if there is epistatic, it could be the inbreeding pressure could be not linear. But I didn't check what is the effect of this quadratic effect of the inbreeding.
Okay. And also there is another nice suggestion with the of course you used a proportion of homozygotes by using the markers, but I think ultimately a very good solution could be to use rounds of homozygosity. So did you have the chance to use this kind of information instead of the basic measure you used?
No, I directly used the proportion of homozygosity, but yes, I'm agreed that it can be used also the round of homozygosity, but I didn't check. It could be interesting also to see what's going on using this coefficient of inbreeding. Okay.
So when it comes to the simulation work, if I understood well, you were using the same individuals as breeders for breeding with inbred and for producing the crossbreeding. I guess that for simplicity, we could assume that we could imply that the same criteria is used. But is it? Can we think that this is real? Does it happen in real situations to use the same individuals while in purebred and in crossbred?
Yeah, I think it is that at least in the French pig breeding scheme they use the same males to produce both the within-breed next generation and the crossbred. Because that we use this scheme. But of course, there is another way to do that, selecting another set of males. But we follow this scheme.
Okay.
Yeah.
Okay. No, it just because for me, I mean, of course, I don't have any quantification on the importance of this kind of differences in the selection criteria, but intuitively for me, the kind of selection you have to do within purebreds could differ because the objectives could be different from the selection you do well for crossbreeding, where maybe in the first it's important to fix some alleles that bring some additive value, but also some alleles that could complement with the other bred. And at the crossbreeding, it's important to combine all those together. So it's not intuitively, it's not exactly the same thing. I don't know if you have any thought about that.
Yeah, I think it could be that you can have different select males to produce crossbreds and different males to produce the next generation within breed with different selection criteria. Yes, I'm agree.
Okay. And then, in your regime with the simulation, so you have these two purebreds that for me seem to be selected on the same trait under similar regimes. Is that the case?
Sorry, I couldn't get you.
So I think this is a question on the on the chapter 3 on the. In your regime, you have these two purebreds, and then within purebreds, I interpret that you seem to be selecting on the same trait under similar regimes. So it's a kind of symmetrical thing. Is that the case?
You mean in the part of the simulation of the two-way crossbreeding, or
yes? Yes, in the case where you have these two purebreds with the crossbred.
It was the same traits in both populations.
Okay, so it's a kind of symmetrical thing. So the selection you were doing in the two purebreds, it's the same selection regime. Is that right? On the same trait? I
mean, it's the same trait, but we consider it like genetically correlated between the three populations. So we think that this is, for example, litter size, but each population has, for example, different variance component, and they are correlated across the three populations. And symmetric depends, for example, for the scheme of selection for crossbred performance, that when the crossbred information is included, then it becomes asymmetric, because I didn't do the reciprocal cross, so there is more information from one side than from the other side.
Okay, I understand. So there is a kind of asymmetry that comes from the pedigree, because one contributes with males and another one with females.
Exactly.
Okay, but would it be in a real situation the case that the two purebreds are selected for the same trait? Wouldn't it be that they are selected for a different set of traits, and they will kind of come together and give a combination, a complementation for a third objective? Wouldn't that be the case?
I think in the real schemes they use specialized breeds. So I think, of course, they work with the same traits, but they have different weight for each trait in depending of the purebred population. Okay.
Then thinking about this mate allocation. So in this simulation. I think what we see is that actually there is there is a lack of benefits from this mating strategy, and well, you give already some interpretation into that, but do you think it's because of well, the simulation conditions that are a little bit narrow and do not exploit completely the conditions where this mate allocation could be useful, or is it just a genuine feature of the mating strategy that is not really performing well? Is it something general or more specific to the simulation scenarios?
I think it will be something general because, for example, if we look to the dominance variance in a F1 population, as the allele frequency from both populations differs, dominance variance will be more lower, I would think. So then we will have less chance of exploiting the dominance effects.
Okay. Okay. In any case, it's not due to the to a problem of precision, because well, when you use the true effects, so it doesn't seem to be something that improves the situation for mate allocation. Well, a little bit, but it's not really very big.
Yes, and the other thing is that across the generation, in the medium and outside, this initial benefit disappears. So because that we see that maybe that by as the parental population differs in allele frequency, so the benefit is becoming less and less and less.
Okay. So I have a few questions concerning this performance of the mate allocation, just to understand well why it doesn't. So do you think that the differentiation between the breeds, for instance, in terms of allele frequencies, could explain somehow this lack of performance?
Yes, I think this could be one of the main reasons that explain that, the differentiation of in allele frequencies.
Okay, and then well, so the logical thing is that well, we saw that there are some slight differences in homozygosity between the different scenarios. So the S1, S2, S3, S4, etc. Both at the purebred and crossbred levels. And I was wondering because, well, behind homozygosity, well, there are other things that are important. For instance, if we can look at what's going on in terms of the favorable alleles. So do you have an idea whether this homozygosity was affecting more or less the favorable alleles or not? Because that could give us a kind of clue about how the different scenarios were fixing efficiently these favorable alleles or not.
You mean if I check exactly the favorite alleles within purebreds or in the crossbred population? Sorry.
Well, in both, yeah.
In both. No, I honestly didn't check the favorite alleles within purebreds and crossbred. I didn't check if it is become accumulated in both parental population. I didn't check.
Okay.
It's one expectation because, for example, I didn't check, but there is some previous studies in the literature that showing this that by doing, for example, selection for crossbred performance, it will be accumulate. Will be first in the crossbred we will fix the opposite alleles, and then in the purebred population, for example, it could be that the same allele will be fixed, but these alleles are not favored within purebred populations. I mean, there is some studies that show that, but I didn't check in my simulation.
Okay, and so in your simulation, of course, the role given to mate allocation was at the level of the crossbreeding, but it was imagined that in order to optimize the whole thing, could be interesting to look at what kind of selection and mating scenario strategy you have to do within purebreds and between purebreds in the crossbreeding level, because in a way, what you do at the purebred will have an influence on where you are going to get at the crossbreeding level a few generations ahead. So it's a kind of let's say global optimization, and maybe it's because of this that this mate allocation is not getting maybe the full advantage of this complementation between breds. That I don't know if you see what I mean is that maybe the shim is too rigid, too fixed, so that the mate allocation is not getting the full advantage of the differentiation between the breds.
I'm a bit lost in your question, but you mean if doing I mean the selection criteria of the purebred, of course, would have some consequence, or will result in something in the crossbred animal. That is true. But for example, we can do after selection of the purebred individuals within purebred. It is possible to do a mate allocation within the purebreds. For example, to prevent inbreeding or to minimize inbreeding within the purebred population, and at the same time we can use the boars or the purebred animals to produce the crossbred with another mate allocation. That in my simulation I didn't observe any improvement.
Okay.
Mate allocation in crossbred, but I didn't apply a mate allocation within purebred, so it could be another scenario possible to check.
Okay. Well, I look at the time. I don't know how much time I have left.
So one or two more questions.
Okay. So so also in this simulation, when we looked at the gain you are getting at crossbreeding level, so it's a very high gain, of course, when you combine crossbred and purebred information. But then when you look at the gain you are getting at purebred level, so it's quite low when you use crossbred information. And so I was wondering, when you get such a low level of gain, you are given the chances to drift to make its work. And so I was wondering to what extent, maybe you have some ideas, or by looking at the results of the simulation, to what extent this lower level of gain at the purebred level could have led to some drift or loss of favorable alleles. I think you have some kind of suggestion somehow but I don't remember now exactly where. But did you get a kind of a clue about that? Is there any kind of drift going on at the purebred level when you select on the crossbreeding level?
That we observed this is the opposite. For example, when we are using crossbred information to select the purebred animals, then we will have a correlated response in the purebred. And of course, if the genetic correlation is low, then selecting for crossbred performance will result in a low response in the purebred animals. So, and yes, this is why we observe, I can say to you.
Okay. Now, is that in the sense that, well, if there was any loss of diversity going on at the purebred level, this will have some consequences later on? Because, well, if you are losing favorable alleles at the purebred level, so those won't be available for crossbreeding later on. But, well, it's just that my okay.
Yes, it is possible that we will lose the favorable alleles to improve purebred performance by selecting for crossbred performance. Yes.
Okay. Maybe I have time for one or two more questions, or one question?
Yes.
Okay. So Yes, you had a suggestion on the use of because of this inbreeding building up at the purebred level. You have the suggestion to use minimum co-ancestry mating, for instance, as a kind of solution instead of the random mating regime that was actually used. And I was wondering, is that really the best adapted method for an hybrid regime? Do we want to promote heterozygosity at the level of the purebred? I'm wondering.
Yes, I propose it like one way to prevent inbreeding, but
if you like, I could try to turn the question on another way. If we promote somehow heterozygosity at the purebred level, aren't we, let's say, eroding the possibility to get extra heterozygosity when we do crossbreeding?
By question, no. You mean with the increasing the heterozygosity reaching purebreds will result in more heterozygosity in crossbreds? That is your point?
Yeah.
I don't think so, because our different traits. So if you improve heterozygosity in purebred animals, but then you are crossing to another unrelated breeds, you already have higher heterozygosis by crossing different breeds. Then what you do within the purebred for increase the heterozygosity, I don't think we have an improvement in crossbred performance.
Okay. Maybe a very last short question, if I have the time, or President?
One short question.
One short, yes. So this is a more general question. So in this new modeling, so it's based on the use of relationship matrices at individual levels, so a G matrix, a T matrix, etc., where the connection to the sequence polymorphism remains an intermediate step when building up the matrices from the genotypic matrices, these Z matrices, for instance. And so in a context where functional genomics and qualitative genetics try to integrate their views, how these kind of approaches could be adapted to accept part of the biological information at the marker level. I don't know if you get what I'm meaning.
Sorry, you mean working with sequencings of the?
Yeah, well, in a way, you deal with relationship matrices. For instance, this G matrix, this D matrix. These are n by n, n being the number of individuals. So, but somehow with functional genomics, we want to stick some kind of biological meaning also to prediction. And so, how do you see the way to use that biological information into this kind of modeling that is based on relationship matrices? Have you thought about it, or did you have a clue about how to deal with it?
Honestly, I don't have too much. I don't know too much about that, but I think if you have some specific functional effects, it can be included. For example, like fixed effects, or depending the kind of the effects that we are interested to include in the model.
Okay. Well, thank you very much.
Thank you, Leopoldo.
Okay, thank you, Leopoldo. So I will now ask some additional question. First of all, I would like to say that I liked reading your thesis, which I found it very, very well very well written. It was not straightforward to talk about pig and I don't remember the word for maize, but in the same thesis. But you did it well and succeeded in showing the differences and the common points to the two species, which was quite fine. I like the kind also of presentation with appendixes, so which explain, which gave extra explanations. I would have liked some introductions, short introductions to the chapters to explain why these chapters and how it integrated in the overall manuscript. But on the whole, the manuscript was quite nice to read. I'm not at all a specialist of genomic selection, so probably my question will be a little bit trivial or naive, but I will ask them. The first one is I'm always wondering why this, when you have dominance, you do not have systematically better fit to the data or better predictive ability when you integrate dominance in your model. Do you have explanations for that?
Depends, for example, in animal breeding, for example, we are interested more in predict the breeding values. So
yes,
you have already account for additive models that include only additive effects. I think are good to estimate the breeding values. But for example, if you want to predict the total genetic value, and in this case, we'll be interested to include dominance. But in general, for us, animals are selected based on breeding values. It is. I would say it is.
Do you think it's necessarily the case for? I agree with cattle, for instance, but in pigs. Are we really interested in breeding values more than in total genetic value?
No, in pig breeding, in the case, they are interested in the breeding value to improve the generation, improve the population within each pre-breed, but also they are interested in improve the crossbred performance, but at the crossbred level, I think.
Mm-hmm. Okay, you had to deal with a set of data in your landrace population, which was quite limited because the pig breeders are quite reluctant to genotype their animals still now. What are the limits in your study related to this limited population size and the fact that probably the sample you are using is somewhat biased because genotypes are not made at random?
Yes, this is something that we have to deal with it because this is the information that is available, because that we try to use to fit the model with pedigree relationship to have some comparison and see that the estimated of, for example, of genetic variance components were more or less close to the obtained with pedigree. So we use pedigree as a reference to see if this sample of the genotyped animals will be close to the estimated of pedigree model.
Okay, if you had no limitation, what would be the ideal data set you would have liked to have?
It will be a random population. I mean, and some higher number of animals or sample, but in this case, normally they genotype selection candidates that already are good animals. So yes, it could be better to use only candidate selection or a higher size of the animal population to better estimate the bias component.
Okay, and if you would you tell the breeding organizations in charge of a breeding program to genotype much more animals, and what animals should they genotype in as a priority?
Yes, I think that they are doing is okay, but they can increase the number of sample of genotyped animals, because in general they are doing first a preselection based on pedigree, and then they genotype the preselected animals. So increasing the number of genotyped animals, I would say will be better to estimate variance components.
Yes, and what type of animal would you increase? Purebred animals or include genotype crossbred animals?
Yeah, including crossbred animals if they are interested in select for crossbred performance, but it is difficult to have all the even purebred animals. It is difficult to have genotyped, and crossbred is even more difficult. But it can be a sample of crossbred population if we are interested in estimating breeding value for crossbred performance. A sample that we can use to estimate the effects, marker effect, for example, and we can use for several generations to do not genotype frequently each generation the animals. So a sample of crossbred animals could be interesting to use for best performance.
For estimating variability, or also for selection purposes?
Yeah, I mean, depending if they still selecting for purebred performance, so we don't need the genotype the crossbred animals. But if they want to try to, for example, do a combined index to balance the genetic gain in purebred and crossbred, it could be interesting to have a sample of crossbred animals genotyped.
Okay, but well, they are selecting on breeding values, but is this the best thing to do necessarily?
To estimate breeding values for purebred performance, I think we don't need the genotype of the crossbred animals. Of course, we will need more information at the purebred level.
Yes, but my question is that for pig breeders, I mean, do they need to necessarily to maximize the purebred genetic trend, or wouldn't it be better to maximize genetic trend at the crossbred level?
Sorry, I'm lost at the moment with this question.
You don't understand it?
Yeah, I'm quite lost in this in the question at this moment.
Oh, the question was to know whether, well, the final goal is to improve the level at the performance at the commercial level. So you could think about increasing genetic gain at the commercial level directly, or which.
I will say that we have to note also which is the genetic correlation between purebred and crossbred performance, because if it is, for example, higher than 0.8, then we just need to select the purebred animals for purebred performance, and we will have, I would say, fashionable gain in crossbred level.
Yes, but if it's lower than that?
So it is lower. So in this case, it will be interesting to have some sample of crossbred animals to include in the genomic evaluation for crossbred performance.
Well, I think it would be, we have to discuss in front of each other. I will move to the simulation study. You indicated in your thesis that the peak breeding schemes were generally free breed crossbreeding system, and you are simulating to breed the crossbreeding system, probably for simplicity, but do you think that the results would have been different with free breed, higher level crossbreeding system?
I think the results for using the selection criteria for crossbred performance will be similar if we work in a three-way cross, for example. But in the case of mate allocation, I think also will be depend on the variance component of the traits. For example, in this case, will be for production traits, considering a three-way cross. But as I check with the different ratio of dominance, even with higher ratio of dominance, mate allocation didn't work. So I don't would expect too much gain also to improving the crossbred performance with mate allocation in a three-way cross. But the results by selecting for crossbred performance, yeah, I think it could be similar that the one I simulate in the two-way crossbreeding.
Okay, if I understand well, your two population came from the same base population.
Yes.
This is, in my opinion, this is a very strong hypothesis. Can you comment on this?
I think in this case it's a maternal scheme, and the two breeds are, I will say, good for maternal traits.
Mm-hmm.
I don't think that it is too strange that come from different, and also I separate for several generations to separate the breeds also.
Okay, so you think that this is not the results wouldn't have changed if you had different origins?
For example, that we did is that I checked the additive relationship between both breeds and also the linkage disequilibrium to be close to the real one, at least to have similar to the simulation, similar to the structure of the real ones, for example.
Okay. Another hypothesis was the population size. The population size you choose, 200 females and 12 males, is quite small for I would say a dam population, which means that inbreeding is likely to increase quite quickly in this kind of population. Why did you choose this population size?
Yeah, first is because to simplify it and not be complex in the computation time, but also we try to follow the scheme that is used in the real ones, I would say, but a proportion of the real one, of course, to keep more or less the same selection intensity for males and females. So we adapted this to try to keep this selection intensity in males and females. But yes, it's quite low, of course. But
yes. Another thing which surprised me a lot is the very low value for dominance variance you use. You have indication that these are real values in the literature? Or...
Yes, actually we used the variance component that were estimated in real Landra's and Yorkshire's population, and it was I would say the extreme case, the case 1 that used these values, and because we tried another levels of dominance to see also how meta-allocation respond to this change in dominance variance.
Yes, can you? I didn't understand very well how you change this ratio of dominance in your study. Can you explain a little bit more precisely?
Yes, I use the variance component from Sham that I told you.
Yes.
But that I changed was the residual variance, the residual variance to adapt to the heritability and the proportion of dominance. And also I changed in one the scenarios the proportion, the dominance variance. So yes, what I varied is the residual variance and the dominance variance. I keep the same values for the additive variance from the paper from Shang et al. 2016.
Oh, you kept constant the additive variance, but you changed the dominance variance also.
Yes, I changed dominance variance and of course the residual variance.
Okay. That's one was very clear to me. If you had, because when you make a simulation, you have to make a lot of hypotheses. So you have made a very, very important job in your thesis, which is quite impressive. But you always want to do, in an ideal situation, to do extra simulations. If you had to time to make additional simulations, what will you do to improve your sets of results?
I would like to try, for example, using a combined index with between pure width to balance the genetic gain between pure width and crossbred performance. It could be interesting. Another thing that could be interesting is in this simulation I work only with genotyped animals, but of course in real we have many animals that don't have genotypes. So another interesting thing could be try another approach like single step, for example, to see how it performs in the medium and long term. Could be interesting in this with this simulation.
Okay, so you were working on two quite different species. You gave some explanation or arguments for interests of combining results on these two species. Do you have other arguments for this kind of studies?
Sorry, I couldn't get its argument for.
Apart from the arguments you put in your thesis about the interest of studying both species, which is mainly on the theoretical aspect of your thesis, would you have extra arguments to justify to a larger extent the fact of considering these two species in the same thesis?
Yeah, at least the main point is that in both species they use crossbreeding. So it is the most that has in common to work with these two species, with pigs and males. I think this is the main argument. This is the relationship between the both fields.
Yes. Do you think it's a good it's a good experience. And would you advise animal breeding students, for instance, to be more interested in some plant breeding schemes, or?
Yeah, I will honestly, I will recommend it. Is good. This study is one example, and we get very interesting results working with animal breeders and plant breeders. For example, the study that was carried out in maize, we use many things. We unify many concepts from animal breeding. For example, we take the concept of include heterozygosity or genomic breeding in the models to account for directional dominance in hybrids, and also the permanent effects that is used in animal breeding. It's interesting. I recommend, but I will say that I don't know if I recommend the student try to, because it also gives a more broad perspective of the quantitative genetics when you work in both fields.
Okay, so I will start by questions now, and I will give a room for Libertat now, if you want to say a few words, Libertat. No, she's not there. We can't hear you, Libertat. No, we don't hear anything.
No, she's trying to connect, but she has a problem with the microphone.
There's a problem with the microphone? Okay. Do we wait for her, or do you want to start, Zulma?
I can do that? Yeah, okay. Probably you can tell her that disconnect and connect again. Yeah. So, I don't have questions, but I want to say something about David. And working with David during these 3 years has been a pleasure, because you are a guy that is, you are enthusiastic, you are curious, dynamic, independent, and always you are very optimistic. And that is very nice to work with somebody like this. Can you hear me?
Camera.
Well, because David can't see me.
Yes.
Okay, sorry. And thank you very much for this beautiful experience, for all the discussion about the plant genetics. I learned a lot of things about maize, and thank you very much for the great guacamole that you prepare in all our parties. So thank you, David, and all the best for the future. And I hope to see you in Guatemala to see Tikal, the Maya city that is one of my dreams that I want to visit, and it will be a pleasure to have you as a guide.
Thank you. It will be my pleasure to have you in Guatemala.
Thank you.
Okay, thank you, Zulma. I don't know if Libertat succeeded to...
Liber, are you there?
No? No, she doesn't seem to be there.
No, probably she has a connection problem, but she can say something after.
Okay, so we will move on now. So now we have deliberation time. So we have technically to have to do something which is not straightforward for me, which is to isolate the jury from the rest of the connected people. Andres, how can we do that?
Well, what I propose is, so it's, how long will it take? Half an hour? 10 minutes?
Maybe less than that, I don't know.
So what I propose is that everyone disconnect except people that is in the jury. So everyone else leaves, and you connect back at 4 p.m. 45, and I will keep everyone outside now.
Okay, we'll do it this way.
I will be like a policeman, and I will also leave the room. So we'll just check that people is not checking.
Okay.
So I leave?
Yes.
Yes, we
I
leave.
think that
Me
we
too.
have to leave also, David.
So, okay, Catherine, Fernando, Hélène, Laurence, okay, Alain, Audrey, can you guys leave and you connect back in 20 minutes? Okay. David and I will also leave the room.